5 prime o 5 prime.
Hindi ka sa akin.
O, dali na. You should know the complementarity, ha?
Gusto mo ako? Hirapan ka ba?
Oh, sige. Now, if there's a presence of 9 mer, assuming ahead of this strand may 9 mer, sequence of adenine, thymine, adenine, thymine. Okay? So, the DNA... Sana? Bakit ganun yung A? Dapat small letter lang. Okay. The DNA A will bind to it. Okay? Of course, with the use of ATP, which is hydrolyzed, so that the helicase in the form of DNA will cut the sugar phosphate bond, forming the replication bubble, and it also facilitates the binding of DNA B and DNA C, and extend the unzipping, forming the replication foil. Okay. Next, anong susunod? Uy, anong susunod? Saan yung ano nyo? Ma'am, unwind na nga eh.
Priming.
Priming. So, ano yung priming? The DNA polymerase C cannot bind to the 3' end because it requires a free 3' hydroxyl end. So, it requires a primer. Lagay nyo sa in front. Catalyzed by the RNA primase, so that the DNA polymerase 3 can bind to it and add nucleotides. So if this is adenine, if this is guanine, you put the complementary base. Okay. And then now there is a simultaneous replication. So whatever happening on the first strand also happens on the second strand. So again, if this is 5', yung end part niya is 3', ha? Therefore, the DNA polymerase 3 cannot bind to it. Again, it requires what? The primer, which is catalyzed by the RNA primase. And then, the DNA polymerase 3 can bind to it and add nucleotide. O, ilagay mo. Ilagay mo complementary base. Ay, sus. Baliktad. So, cytosine is to guanine. Okay. So, there is again the unzipping of the helix. So, since there is no exposed 3' end, so there is a continuous synthesis of the nucleotide, and then put the complementary base. Cytosine is to guanine, thymine is to adenine. How about on the other strand? Ano mangyayari? So again, kasi 5', 3' end, so there's an exposed 3' end. Therefore, the DNA polymerase cannot bind to it. It requires again another primer, which is catalyzed by the RNA primase, and that's the time that the DNA polymerase can bind to it. So you put the complementary base. So adenine is to thymine. Okay. So you unzip again. Tatlong unzipping lang, maliit ang zipper natin. Okay. So this is now the leading strand. Why? Because there is a continuous synthesis of the nucleotide or the DNA. So you put the complementary base. Guanine is to cytosine. Kabisaduhin nyo yung color coding natin. Cytosine is to guanine. Thymine, adenine, and thymine. Okay? How about on the other strand? Sa kabila, ano? Sa kabila. Uy, ano na? Nakatingin lahat. Okay, again, it requires the RNA primer because there is no exposed, because there is no free 3' hydroxyl end. and RNA primer is catalyzed by the RNA primase, and then the DNA polymerase III can add nucleotide. O lagyan niyo yung complementary base. Okay. And then, anong sunod? Ano yung bakit? Ano yung tanong mong bakit? Primer yan eh. Gusto ko. Anong susunod? Proofread. Okay. Ano gagawin? So the DNA polymerase 1 can act, it runs from 3' to 5', so it will proofread and remove erroneous mismatch against nucleotide. Okay. Sabi niya bakit daw yon. So it will remove the mismatch and then put the complementary base. So if this is guanine, cytosine, the complementary base is guanine. Tingnan niyo pa kung may mali. Okay, it also remove this part, so it will be replaced by thymine. Okay, meron pa ba? Tignan nyo kung meron pa. May primer kasi.
Wala talaga. May primer din.
So this is now called what? Anong tawag dito sa strand na to? So this is lagi strand because there's discontinuous and there's now the formation of Okazaki fragment. Okay. Or RNA removal, primer removal. Ano na? So the DNA polymerase 1 will again runs from 3' to 5'. It removes the primer and fills the gap. Pag sinabing fills the gap, put the complementary base. So cytosine is to guanine. Okay. Remove the primer. Okay, remove the primer. Okay. Thymine. And then remove the primer. Okay. Yung primer ha, dinadigest niya, excise and digest, hindi lang itatapon. Dinadigest ng DNA polymerase 1. So, what happened now? Replication is completed and the DNA ligase will seal the gap between nucleotide. Asan ang ligase? Ligase. Okay, hindi ko na ano yung gyrase ha, pagka nagkaroon ng supercoiling. And then the SSBP, if there is now the open conformation, okay. So the byproduct now is, wag nyo nang tanggalin, but actually, maghihiwalay yan. And the byproduct is what? One old, one new strand, one old, one new strand, and the mode of replication is semi-conservative. Okay. Tapos.
Para same po yung SS.
To stabilize the open conformation pagka nag-open. Yes, walang portion. Sasabihin lahat. Oh, sige.
Sadya ka ng mali. Ganyan talagang sadya ng mali.
Ay, hindi. Depende sa'yo kung gusto mo. Kung ayaw mo naman, you just mention.
Feeling na okay dito, tama naman lahat yun.
Oh, kanino to? Oh, practice. Hirang lang to, ha? Oh, panganin pa.